Okay. English, Hebrew? English. Okay. I actually had a little Hebrew start, political start, but I'll do it later maybe, because we have to do something about what happens in this country. It's really something you, as a young generation, should be active on, but I'll do it later on in Hebrew. So I'll start with the science, and then I'll go to the politics a little bit, very little, and then will be again a science. But I'm looking for something that I think I lost. My little pointer. Okay, so we have three meetings together. This time you are not going to present. I'm not asking you to present any papers, so it's only me. And my role here, so to speak, is to try to give you intuitions. I'll do a little bit math here and there, but mostly it will be intuition rather than detailed the development of equations. So the main thing is intuition. And
point that logical.
Sorry?
Can you point the light?
Yeah.
The thing, okay. It was on me, so I will. And I want to focus basically on computation at the level of single neurons. One thing I don't know, and you should, along my talk, correct me or enhance me or change, because I don't know exactly what you know. I know that some of you studied some aspect and some not, maybe. So tell me if this is already understood, then it will help me to go further. And if not, then I'll do my program. So it should be very interactive and very informal. And please don't hesitate to ask questions, really, any question. Because as I said, I want to really develop intuitions about these neurons, which are, as you will see, becoming more and more, on the one hand, more and more, we know them better, more intimately, in terms of experimental aspects. We still do not completely understand this very sophisticated microchip in terms of its input-output plastic properties. We know some, but we don't really completely understand this very fundamental building block in terms of what it gets as an input while I behave, and what type of output does it generate. And of course, without understanding the microchip, which is very sophisticated, not a point neuron, but rather a complicated neuron, we are not going to understand information processing in the brain. So I want to introduce to you what we do understand and what we don't understand, where is the missing points, what are the new directions, including machine learning, into the neuron, so to speak, and so let's see how we progress. But this is the plan, and we shall see out of this plan what I can do in 3 times of meetings. So I want to introduce the neuron very briefly. You know some of it, but I want to start with the history. We cannot do without a little bit of history. and then I want to speak a little bit, very little, because you see now really every day new information about places in the neuron that we could not recall from. The thin branches in the dendrites and the thin branches in the axons were unknown to us, but because of new optical and other technologies, we started to better understand these remote places or thin places. So because typically we knew everything, so to speak, from the cell body, which is only one point out of this very distributed system. So I want to introduce to you these key new findings about the neuron. Then I'll start to develop the passive cable theory, very brief, again without the details, because this is the fundamental of our understanding, the fundamental mathematical theory for understanding neurons as an information processing input-output devices. I will say just a word about why do we want to model the neuron. For you it will not be very essential, because you understand that the whole philosophy of this center is that you are not going to understand anything without modeling. You can describe things in words, but eventually it should be somehow formalized in a theory. So I will say a few words about that. I'll summarize with the main insights from cable theory, passive cable theory. Then you can see this word, of course, is an approximation, because we know that the neuron, the dendritic tree, the input side of neurons is not passive. And that's the whole thing, that it's a very highly nonlinear device, not only because it has a spike in the axon, also because it has nonlinearities in the dendrite, And these nonlinearities we know today are essential for the computational capability of the cell. So I'll speak about the nonlinearities, and then I'll summarize with the computational ideas. It's not so easy to, of course, show that when some animal is doing some behavior, this is the computation that the neuron is performing now. But we have some ideas, some beginning of some experimental confirmation of some of these ideas. So this will be the end target of these little three lecture courses. Okay, let's start. So I cannot start, of course, without these two guys that you know, I'm sure, I hope. And they got the first Nobel Prize in neuroscience. So the Nobel Prize was started in 1901, but the first Nobel Prize for brain aspects was Golgi and Cajal, Camilo Golgi and Cajal, the big enemies. They are enemies because they didn't agree on some fundamental aspects about the neuron. We'll discuss it in a second. But anyway, absolutely both were amazing anatomists. There was never since then anatomists at that scale. Of course, the anatomy was very, very, very fundamental relative to what we are trying to do today. But they were really the one who reintroduced systematic anatomy in order to characterize at least the basic structure, the basic elements in the brain. They got the Nobel Prize, and as you may know, on the podium they fought between each other because of this disagreement that I will talk about. So this was it's very interesting, I don't know, from the history of science to see two great men, scientists, do not agree with each other, but still both get Nobel Prize for the same thing. And so this was the real character, this Ramon y Cajal, in many ways, a very, very unusual character, developing or enhancing the technology that Golgi developed. So Golgi has a technique that is called the Golgi staining technique. It's staining neurons. We should talk a little bit about that in a second. He started to develop a method that enabled to stain individual neurons very, very sparsely. So here a neuron is still today we are using it, but an old staining technique. But he stained and into the network he poured this stain and he developed it like in a film. And then he got this nerve cell is stained and this nerve cell is some are stained. Many of them, most of them are not. And this was his luck because then he started to see the individual ones. Not that we did not know that there are elements in the nervous system, but not to the level that these two people reconstructed them and studied them with this old microscope. And as I said, Ramon y Cajal had this graphical mania. It was like a mania because he was painting all day long neurons and neurons and neurons, and he has books and books and books about beautiful drawings, and which you can find in the Cajal Museum in Madrid today. Really wonderful paintings. And this mania, it's not only the mania of graphical mania, but also his insights, as you will see in a second, gave us some new intuitions about what are these neurons doing, how do they operate. And of course, at the same time, there was another mania the same years by Van Gogh. So of course, I don't think that things are not connected in some sense, although maybe they didn't know about each other. But of course, Van Gogh had the same mania. So these are the important years of beginning of drawing trees to this level, both by Ramon y Cajal and Golgi and Van Gogh in Arles before he cut his ear. And this is the staining of Golgi. So this is a cell. This is a pyramidal cell, a layer 2-3 pyramidal cell. We'll talk about it. This is original staining by Ramon y Cajal using Golgi staining. And you can see quite well at this level of resolution, but it's much better, of course, if you go to the museum. You can see really the cell body, you can see dendrites, you can even see spines. We should talk a lot about dendritic spines. You can see the axon of layer 2-3 in this case. And Ramon y Cajal, I don't know if you know, but he likes to call these pyramidal cells, which in you are the majority in the cortex, in this case. Ramon y Cajal did every part of the nervous system, the spinal cord, the retina, any part of the brain, he reconstructed cells. So I'm just giving one example of the cortex. He did many, many things in many, many animals, including human cells, the human neurons. We shall talk about human neurons because this is a new revolution that is happening now in the world, starting to study human nerve cells. But we shall talk about this later. So also Cajal started to reconstruct also human neurons from post-mortem, but this is from a mouse in this case, and this is a pyramidal cell, and you can see that he tried to reconstruct it and draw them. I'll show you some drawings. And he used to call these cells the psychic cells. So the original name of Ramon y Cajal for the pyramidal cells, he didn't give the name pyramidal cells, it came later by a German guy, but the name pyramidal cells, which we use today, was not used by Ramon y Cajal. He called them the psychic cells, because he thought, and of course he is right in many ways, that there is this majority of about 80 or 85% of the cells in the cortex are pyramidal, of the pyramidal type, of the psychic type, and he thought that these cells must be very relevant, essential for our high-level capabilities of mammals. Of course, these are not only human. So all these new capabilities of mammals are 200 million years old, and cortex is 200 million years old, so the evolution of cortex with pyramidal cells brought new capabilities to this breed, to this species that it calls mammalians. We are, of course, very sophisticated mammalians who believe that we can understand ourselves as brain researchers. Not other mammals, not many other mammals explore their own brain or brain of others. So you can see some of his wonderful, really wonderful paintings. You understand that he was looking at the microscope and at the same time he drew what he sees. So he was looking here and he was drawing there, and this is not so easy kind of visual motor capabilities, but he did it in such a wonderful, really wonderful way. As you can see, this is cerebellum, Purkinje cells, and you can understand that it's all handmade, really. And this is very classical neurons. So every book, this is the most well-known neuron in the world. Yes, this pyramidal, this cerebellar Purkinje cells. You can see the cortex with interneurons and pyramidal neurons, the different layers. You can see axons coming from the thalamus. All this is again painted many hours every day by Ramon y Cajal. That's his mania. Which is a very interesting mania. He really liked to draw, but he did it in such a wonderful way, really. And this is a drawing from the retina. So you can see that he reconstruct the retina, cell types, ganglion cells, bipolar cells, and so forth. You can see that he touched every part of the nervous system and he tried to understand the circuits. So this is an anatomical, so to speak, understanding, but of course it's essential in order to better understand later what kind of signals are going on there and so forth. So this is really the beginning of modern neuroscience. Modern neuroscience is very young, 120 years old or so, and this is the beginning. But you can see that this is very sparse. By the way, when I was recently in Madrid with a, so to speak, grandchild of Ramon y Cajal, so it's Javier de Felipe, is another great anatomist, probably the best anatomist today, living in Spain, not coincidentally. Not that he met Cajal, but he was inspired by Cajal and he's Spaniard, so there is, of course, historical aspect to, you know, which country, for example, the Hungarians are also very good in anatomy, so there is traditions, and so this Javier de Felipe, on his walls, there are the whole room, the whole apartment, the whole flat is full of the original prints. You know, the prints were done for the paper, for the printing was done on copper, you know, etching on copper, and here's the original coppers, and all these walls are painted with that. And he told me something I did not know half a year ago, although I read a lot about Cajal, but I did not know that this, for example, a drawing like this that you see now is not something that he saw at once. Okay, at once he saw this cell, let's say, so he reconstructed this cell. And then in another slice, in another preparation, he saw, let's say, this cell, so he put them aside. So it's a composition. It's a composition of many slices. So this is a reconstruction of a, let's say, generic cortex. It's not a cortex of one animal. And of course it's very sparse because of course there are many other cells. And it's a very small percentage of the cell, luckily, that are stayed, as I said, that enables him to see individual cells during this process of anatomical reconstruction. You know that today, of course, one of the biggest projects today is the connectomics project. We don't have it in Israel. There is one group in Europe, in Frankfurt, that is doing connectomics. In this case, it's an EM reconstruction, full reconstruction, unlike this light microscope reconstruction. And this is an EM reconstruction, and we still today don't have even a cubic millimeter fully reconstructed from the cortex. So even today, with all the sophistication of computer vision and automatic reconstructions and all that, we don't have a cubic millimeter, it's very small, cubic millimeter of cortex. But we will have within a few years a cubic millimeter of all the nerve cells, all the synapses, who is connected to who, numbers and all that. We don't have yet that. But I'll tell you later, if we come to the Blue Brain and Human Brain project, where we start to do something similar to in reconstruction, but much more detailed, with some sampling of the nervous system to try to know more about the cubic millimeter of cortex. Because otherwise, of course, we will not be able to understand how signals flow in this cubic millimeter, how inhibition affects excitation, type of cells, when are they active, and so on. Anyway, so this is the beginning, and you can see that Harmonica Hall already marked cells, body, and axons by some kind of letters and so forth. And you can see these really beautiful reconstructions that he did.
Yeah. And when you talk about the cubic millimeter, what's the resolution?
Of this one?
No,
or
in the future one.
So the EM, okay, so that's
I
a
mean,
good.
do you mean like all the things within the cell?
Sorry, no, okay. So this is light microscope. So until today, people reconstruct with light microscope pieces. The light microscope resolution is on the order of, let's say, maximum, with a very, very good optics, very sophisticated cleaning of noise. You can get by the physics of the light, you can get something, let's say, maximal tenth of a micron resolution. So at the level of 1 micron, which is less, for example, than the level of a synapse. Synapse is smaller than tenth of a micron. So you cannot see synapses with light microscope, as they are. You can stain today's synapses, but because they have particular proteins, and so you can see these puncta, these points. You can say there is a synapse here, there is a synapse there, but it's not at the resolution of really the structure of the synapse. So for the structure of the synapse, you need light electron microscope. So you need to cut very, very thin slices. This is at a nanometer scale, so thousand more than the light microscope scale, and then you get everything, so to speak. When I say everything, I mean at the level of nanometers. 20 nanometers is the best EM microscope. So of course you don't see everything. You don't see the nucleus or the details. That's an even higher resolution. But I'm speaking about connectivity, so I'm speaking about synapses. And this you can only see with EM. So that's what I meant connectomics. So the connectomics project is at the level of synapses, which is 25 nanometers. So when I say you see everything, I mean you see cell types, and you see all the connections physically using this EM microscope. The thing is that you have to cut very thin slices, let's say 10,000 slices at the depth of the cortex. By the way, the depth of the mouse cortex is, let's say, 2 millimeters. So from here, from the pia to the depth to layer 6 is 2 millimeter. In our case, it's 3 millimeter. We are thicker than the mouse cortex. And so to reconstruct this, you need maybe 10,000 slices with EM, and then you need to reconstruct it one on top of the other, and nobody still succeeded to do it. Some people are against it, of course. Let's say physicists are against the whole project of connectomics, or it used to be. Chaim Sokolinski was against it because this is, like, first of all, it's terrible work. I mean, it's hard work, black work. It's not good work. But the question is, do you need it? Or is it enough to sample and say, okay, I have some principles and I don't need all the details and so forth. But today more and more people feel that this is an essential part, at least to get some statistics to the details of a whole, and it's only 1 cubic millimeter, certainly not the whole brain at the level of EM. So we don't know a lot about the anatomy of the brain. This anatomy is dynamic. We should talk about plasticity. New synapses are created, so I'm catching the brain now, but in some 2 minutes later, it will be a little bit different brain. So what do we do about that? But we'll discuss it later. This is only an introduction, of course. One important thing is that Cajal, as I said, had, and besides having reconstructed neurons and so forth, he added these arrows in some of his work, and this is his original arrows. So this is his imagination. You can see that he imagined arrows. So there are no arrows in the brain, I can tell you, at least we didn't find them. But there are connections, and he didn't see the connections. He didn't see them because of what I said about the resolution. Still, just because of the anatomy, seeing an axon coming from the thalamus, which is, he knew that it's an earlier stage from the periphery. So let's say it's a visual cortex, there is an eye, there is the LGN, thalamic part of the visual system, and then there is the cortex. So this is coming from the periphery, so to speak. So he said there must be some signal. He didn't know about spikes, or he knew there must be some electrical activity. So the brain was electrical, but he didn't know anything about direction. So he said there should be some flow of information, probably electrical. Then there is some kind of a jump between the axon and the dendrite. What is this jump? We don't know, because there is no, we didn't know what's happening here. And then he said that there is a signal flow in this direction from the tip of the dendrites towards the soma, and from the soma to the axon of this cell, and then from the axon to the dendrite of the other cell. So the whole notion of what he used to call the neuron doctrine, and in particular the polarization theory, he used to call it, the dynamic polarization theory, is that cells are polarized. They are polarized in the dendrite by some input. They are polarized in the dendrite, and then there is a flow of this polarization, of this electrical signal somehow from the dendrite to the soma and so forth. So that was his idea about signal flow. And we still think today about this like that. Of course, it's not the full story. Today we know that some signals, I'm sure you heard about the backpropagating action potential, for example. We know that the spike that is initiated here can backpropagate from the soma back into the dendrite, so these arrows are not correct. There is also a back arrow. This, of course, we did not know. But this is called the theory of dynamic polarization, these arrows, and the notion that there are individual cells is called the neuron doctrine. So the notion, the neuron doctrine, although he did not invent the word neuron, but the neuron doctrine, the fact that there are cells separated in the brain, is his idea. The notion of cell, the fact that any tissue, living tissue, is built from cell, was known from the 18th century already. But the fact that the brain is also built from cells is his idea. And that's exactly what Golgi did not agree on. He said it cannot be. Absolutely no. That the brain is also, like every other tissue, built from individual cells. No. Golgi said no. Why not? Because he didn't see, unlike what I draw here, or what Cajal drew here, he saw many things connected, so to speak, to each other, one big jungle, and he said these jungle are not built from individual cells, but for what he called a syncytium, a continuum. It's a continuum tissue. It's not built from individual cells. So this was the big fight, really. Cajal had the neuron doctrine, saying that these are separate elements, because what he thought he saw in the microscope, and he didn't see that. Of course, there are many other cells here, so maybe everything is connected to everything or not. We know that everything is connected to everything, but indeed there are separate cells. They are connected by synapses, but they are not touching each other. It's not one thing. Golgi was really a romanticist. He was Italian, he believed in opera and so on, and emotions, and so how could emotions, how could feelings, how could consciousness, all these wonderful things that we have is from neurons. Something is not working in his mind. Of course, he said it must be some different tissue, unlike the heart and unlike our muscles. It's a different tissue. It's a continuum. Not that it solves anything, but at least this was the fight between the two of them. So Golgi in his podium, he said, my respectful colleague, anatomist and so forth, is absolutely wrong. He said so in his lecture, the Nobel Prize lecture. It's absolutely wrong, with all respect, because there are no individual cells in the brain. And Golgi said with all respect, and Cajal said with all respect, they are individual cells. But they could not prove it. We could prove it only when we had EM microscope. And when we cut with EM microscope, this was in the middle of the 20th century, we could start to cut and see really cells are not touching each other. Synapses are separated, the cells are not touching. So you can kill one cell without affecting other cells. They are separate. So we know that there are cells, but this was a theory, because how can you know that there are individual cells? This was an invention, a fantasy, which was apparently correct in the case of Ramon y Cajal and incorrect in the case of Golgi. It has, of course, many, many other implications, whether it's one continuum system or whether it's separated units, both in terms of information flow, one directionality. All this directionality is enabled because there are separate elements and so forth. But I don't want to go into the details of history. There's a whole book about that. It's called the Neuron Doctrine. So anybody is interested about the history of the neuron should read the book The Neuron Doctrine by Gordon Shepard. Of course, today we can do much more. For example, this is something that nobody could do before, and many people now could record directly into the dendrites, or so measure electrical activity in the dendrites. You should understand, of course, that the cell, this is a Purkinje cell from this, the nice one that was drawn by Cajal. You should understand, of course, that most of the membrane area, most of the interesting things that are happening in the neuron are happening in the dendrite. Because all these 10,000 synapses, in the case of pyramidal cells, or 100,000 synapses, so this Purkinje cell is the most innervated cell in the world. It has 100,000 synapses, which is 10 times more than an average cell in the cortex. But let's speak about an average number. Let's say 10,000 synapses are distributed on the dendrite. And we didn't know what are the signals that are happening there. Are they all linear, passive, synaptic? There are spikes there, or what is happening in the dendrites? All this is relatively new. In the last 20 years, people succeeded to record in slices, and today even in vivo activity of electrical activity, chemical activity in dendrites. Just to complete the anatomical story, and then we'll go to more physiological aspect, just to tell you the anatomical aspect. I hope you understand, of course, that a single neuron, in this case reconstructed by Kevin Martin, also great anatomist from Switzerland. So if the green thing is the dendrite of pyramidal cell in this case, the red thing is the axon, the local axon. And of course you can see something that maybe sometimes you don't think about this axon. You may think about axon like wires, electrical wires going through a tree, but it's not. It's much, much more complicated. It's a whole tree. So this is the axonal tree, and these yellow points are the presynaptic boutons. That's where the transmitter lies. So you can see that a local axon in the cortex of this layer 5 pyramidal cell is innervating a whole region around it, surrounded by it, making many, many synapses on the order of 10,000 synapses per one axon into its neighboring, and sometimes it can go, of course, to other regions, maybe outside of the cortex to the thalamus back, or to higher level regions in the cortex, and so on. But just I wanted to have an impression that your impression of a neuron will not be of a complicated dendritic tree and a simple axonal tree, but rather two trees of different types. Okay, so this is the introduction to the neuron, which many of you knew, and there are few people today that can do what Matthew Larkin can do, is record at several points. We shall talk about it in a second. At several points in the same neuron. This is a slice. And this is really, you have to have a very special good eye and a special good capability to patch a dendrite here, to patch a dendrite there, to patch the soma, and even some people can patch an axon at the same time. So you can try to understand the input in the dendrite, output of the axon, and the correlation between them by doing these kind of things. But this really requires recording either by electrical electrodes or by optical means, let's say 2-photon microscope, trying to image what's happening here and there. So this high resolution, we'll talk about a breakthrough in my mind that happened a few years ago in the understanding of what kind of signals are running in vivo really in vivo during visual processing in the dendrite, and what did we understand from that. So here it is. So what I showed you was electrical recordings. This was actually the method, which we should speak a little bit about that, just to make sure that you all know about this backpropagating action potential. So this was the method to really see that the signals, that action potential typically really starts here, the sodium action potential. The classical action potential of Hodgkin-Huxley starts here, so to speak, and it goes into the axon, but it also goes backward. We could not know about this without recording in the dendrites while the action potential was initiated here. So this is the work of Greg Stewart and Bert Sackman from 1994, and this was a student of is a student, or was a student of Sackman, and he knows to do these things, to record in the dendrites, both in vivo and in vitro. But the more classical thing today is to do what Arthur Connors did some 7 years ago, and this is really a breakthrough in my mind in terms of understanding the input-output properties of a neuron. So I wanted to highlight this particular paper because I think it's the most interesting in that sense. It's really the breakthrough and the most interesting thing that of course there are many papers after that by Connors, by Eli Nelkin and Connors on the auditory system. So this is a visual system. This is a in vivo recording, so this is a living mouse, anesthetized but living mouse, and you can record from V1 optically, and you can show him here, the mouse, some visual inputs, so grading going this direction, and that direction. And you can record from an individual cell during visual input. Okay, the nice thing about this work is, as I said, it's the first time really ever that the development of this two-photon microscopy was pushed to the resolution, the two-photon microscope, which typically, let's say, AD, Mickey London and other, although they are doing now also dendritic recordings, but typically you record the activity of around the cell body. You know, this cell, this one was activated, this cell was activated by calcium signals. But the question is, can I see the local activity in the dendrites? This is much, much finer resolution, because let's say that the synapse is active, one synapse is now active, and there is a flow of calcium postsynaptically. Into the postsynaptic region of the synapse there is a flow of calcium, because there is an opening of NMDA receptors and so forth. Can I record locally during visual input in the dendrites? So this requires higher resolution of two photons. And that's what Arthur Connors did, and you can see that he tried to record. So this is, as I said, this is a cell in the visual system, and this is, in this case, it's not a pyramidal cell, but rather, I think in this case, no, this is a pyramidal, this is a layer 2-3 pyramidal cell. So in one focal plane, let's say in one focal plane, you have this part of the dendritic tree on the focal lane of the two photon. You know, the two photon has a very thin optical focal plane, let's say a micrometer, and then you have to focus deeper. So if you focus on one focal plane, you will see some dendrites on the focal plane, and some dendrites are deeper or above you. Is it clear? So you are looking at the focal plane, and you can ask, is there a calcium activity here, or calcium activity here at that location? You assume that that location has a synapse, if there is a calcium activity, which may be true or not. But that's what you cannot see the synapse. You can see only the calcium activity following, hopefully, some synaptic activity. So you can see that this particular region, for this particular direction of visual stimuli, there is a calcium activity at point number 4. Let's say this is point number 4. So point number 4, the synapse on the dendrite at location number 4 responds to this direction of visual input. Okay, and another location, let's say this location, let's say location number 7 in the dendrite is sensitive to this direction of motion. So we are talking now about the input itself. I'm not talking about the spiking activity of the soma. I'm talking about the input to the dendrite. So this is the first systematic work trying to characterize what do the dendrites get when you look, or the mouse looks at the world.
So it implies of the tuning curves of the presynaptic cells, maybe.
Exactly.
But what is the
we'll talk about it in a second. You're right. I'm representing by the synapse that I get. Okay, there is a synapse that I look at. I look at optically at the synapse. Where can I find the thing? Oh, here. Okay, so I'm looking at a cell. Let's say this is a pyramidal cell, and some other cell that I don't know the origin of this cell. It sends an axon and makes a synapse here. I don't know who is the presynaptic cell. This I don't know. I just look at the activity in the dendrite of the postsynaptic cell, and I'm trying to characterize the receptive field of this synapse, of the input itself in the dendrite. And I'm saying, as you just said, that this particular synapse, number 4 here, is really sensitive. The presynaptic cell that gives me this input is sensitive to this direction. Less sensitive to this direction, a little bit sensitive to this direction, so not only to this direction, but to the opposite direction. So I can do a receptive field. I can characterize the property of this particular input. This is new. We were not able to do it before this work of Arthur Connors, because he pushed the technology of 2 photons to the level of individual synapses. So one thing that happens, so if you do many synapses in the same cell, so you look at this cell at many locations during visual stimuli, you can see something that is very clear here, that this particular cell in the visual cortex, V1 of the mouse, is getting, of course, many synapses. You don't record from all the 5,000 synapses or whatever that this cell gets, but you record, let's say, from 20 synapses. And you can see that each individual synapse has a different receptive field. Okay, so this synapse is very sensitive to this direction of motion. This other synapse, I don't know, let's say, this synapse is sensitive to this direction of motion. And so you see that this is what is called the salt and pepper organization. Because one cell, it's one neuron, actually receives visual input from all directions, basically. But eventually, the output of this cell is sensitive to one direction. Okay, so I'm sensing the world as a cell. I'm sensing the visual world from all directions. All my synapses, this synapse is active to this direction, this synapse is active to this direction. But eventually, somehow, I compute, we don't know how, I compute some kind of average or majority or some kind of other principle that we don't yet know. The dendrite somehow compute all these inputs and generate an output that is directional selective, let's say to 45 degrees. And we still don't know what is the rule by which this salt and pepper, why we call it salt and pepper, because this is a mix, it's a mix of salt and pepper, but of course not only salt and pepper, but of many ingredients, directions, this and that, and eventually there is an output that is unique to this cell, at least in this mode. Is that clear?
I have a question.
Yes, of course.
How do you know that this neuron has a specific output?
Because at the same time, we really should look at this paper at some point, but at the same time, really a very interesting paper and very influential, not only interesting, but many groups, including Eli Melkin, worked with him to develop this capability in Germany in order to also record from auditory neurons and somatosensory, whisker system and so forth. But this salt and pepper organization is clear to all the system. And then you can also record, and this is maybe in the next slide, I don't know about it, but you can record patch recording, electrical recording from the soma of this cell. So you also have the output of this same cell.
Yes, but maybe it participates in many populations that all cause for different orientation.
It is part of the population.
So it's not like an average, and this neuron codes a specific orientation that is the average of all of them?
This we don't know if it's the average.
It participates in many orientations.
I'm saying this cell samples many orientations. This we know from the input. Okay, so we know that it samples from different orientations. At least from this limited sample, it's only 20 synapses or so. We can reach 100 today of one cell, but not 10,000 or not 5,000. But we know a good sample, let's say. So you're right, it is sampling this orientation, it is sampling another orientation, and so forth. So it samples the world of orientations on the input. And then it generates a particular output. This I know because I have an electrode in the cell body. And I see that as the classical was done, as was done by Huber and Wiesel, I characterize the output selectivity of this cell. And I say, let's say that this is 45 degrees. So I have a receptive field for the output of these cells from my electrode in the cell body, and at the same time I know what is the input to the limited extent. Does that answer your question or no? You had another thing in your mind. Say, what is missing?
I just meant that maybe the input of the neuron is not a specific orientation. Maybe...
No, it's not a specific. The input to the neuron is a combination of orientation.
Sorry, I meant output.
Oh, the output. No, I can record the output. I can do the same. It's the same input I do to the visual world, and I characterize the output as classically done, and I see that it has a particular sensitivity, at least for the recording period. It has a particular. This cell is 45 degrees. It's true, and that's one thing that people might tell you. The capability to become sensitive to another orientation is already there. Let's say that the output of this cell now is 45 degrees. Okay, so I'm doing this. I record the output of this particular cell here, and I see that this is angle here, and I see that this is 45 degrees. That's it. That's the output. But it could be, in principle, because it has all the inputs available, that if I make this synapse more strong or this synapse more strong, it will shift its capability plastically. Because it has already all the inputs. The inputs are there. I don't need to build the system again. I just need to change somehow about the weights of the different orientation and shift this neuron to another orientation. Maybe that's what you meant. But for a given period of time, while the experiment is being done for an hour, let's say, it doesn't change it. But it could be that if you explore the world, if you see only 30 degrees all your time, then the cell shifts to 30. This we don't know. But it has, at least it has the wiring there, or the synapses are there. It's only that somehow, at this moment, the dendrites somehow compute, maybe by majority. Maybe the majority of the cells have this orientation. I don't know if you look here, it's not a good sample, but you see many are pointing here. So maybe this and this and that, and I don't know what. If you average them, you will get the output that you see. But it's too small a sample to say that. So we don't know the principle. I don't know if the principle is majority, or maybe the nearby synapses dictate the output and not the distal synapses, or some nonlinear processes highlight this particular synapse and make it very important and essential for the output. I don't know. I'm just saying that it's a beginning of experimentally sampling both the input of and the output of the same cell. That's what I was trying to say. That's the new jump. In vivo, yes, while you really get the visual input.
And how would you know that?
What?
What's the computation? I mean...
How in principle I would know that? I need a model. First of all, I need more inputs. Yes, I need to sample more inputs, and people are pushing this into, as I said, 100, maybe 1,000. If I will have enough inputs during the visual processing, and I will have the input, and I will have the output, and I will know something about or we'll talk about the nonlinear properties of the dendrites, I hope that my model, if I have a good model of this neuron, I will be able to predict what output will happen, what output will be generated by knowing the input and the cable properties that we should talk about it of the tree. Then I will say that I understood the input-output properties, but I'm not yet there. Because it's not only the synapses, it's also the properties, the electrical properties of the dendrite that shape the output. The output is not only shaped by the synapses, but also by the intermediate properties of the dendrite. So we should talk about models. But that's exactly why we need models. But it could be, in principle, let's say I had all the sampling, and I do a simple averaging, and I say it's just a simple average, or it's just a majority. The majority of synapses dictate the output. That's a simple model. I don't need anything more. But I cannot say that at this point in time, because of the limitation.
Can we destroy some specific presynaptic cells and measure the signal trend?
Yes. Today with optogenetics, you can activate or inactivate some of the presynaptic cells, yes, and see the effect on the output of this cell or the synapses themselves, yes. It is possible. It is possible. Not that I know that many people did it, but you could in principle. We don't, as I said, I don't know what is the presynaptic cell to this synapse. It's somewhere there. I don't know if it's the same layer or it's coming from another layer. I don't know the origin. But in principle, I could start to manipulate the presynaptic network and try to see the input-output effect here. This is, in principle, yes.
From single synapses?
So in principle, if I could, let's say, have a cell here, this is high resolution optogenetics. Suppose I have a cell, I think maybe this is a presynaptic cell, and I shut it off, let's say by Hagerod, by some way to shut it off and see that I change the activity of this synapse, because I can record this synapse and I somehow can shut it off, which is very difficult, of course, to find the origin of this synapse. Very difficult. But in principle, yes, I could manipulate one and look at the synapses and so on. It's very difficult work. But that's what is missing. That's what exactly is missing. Yeah. One second. Let's finish this part. Just to summarize this interesting finding. Not only interesting, but new in the technology. It's new. We understand it. We understand the system. As new technologies come, we know much more about the system. It doesn't mean that we understood it, but at least there is something that we can think about having this. So it could be the organization of synapses on the dendrites could be of different types. For example, it could be of this type, meaning that a given orientation, let's say the blue orientation, let's call the blue orientation, is mapped on one dendrite, and then the cyan orientation and the green orientation and the yellow orientation on separate dendrites. That's not what was found. It could have been also that all the synapses have the same orientation. This is certainly not what was found. But this is what was found. You can get on the same dendrite, on the same piece of the dendrite, two synapses, let's say here, that have different orientation on the same piece of dendrite. That's the salt and pepper is at the level of individual branches. You can see at the same branch, one is salt and another is pepper on the same branch. This is a mouse V1. Nobody did this yet at the cat V1, because in the cat V1, I hope you know from Urban and Wizard, there are columns. The whole column, or most of the column, has the same orientation selectivity for all the cells in the column. In the mouse, there is no visual column. So in the mouse, if you take, in the V1 of the mouse, if you take two nearby cells, they have the same, they have different orientation. So the salt and pepper organization was known already before at the level of the output. We know in mouse V1, the two nearby cells could have completely different orientation. So there is no structural columnar functional, there is no functional structural column in the mouse. There is a structural functional column in the cat. Okay, so if in the cat, if you take two nearby cells, they have probably the same orientation. So we don't know, because nobody did this on the cat, whether in the cat, I would expect that in the cat, most of the synapses impinging on the neuron will have the same orientation, because they get this input from nearby cells, and the nearby cells are similar to you in terms of orientation selectivity. So probably the synapses are also, most of them, from the same input. But I'm saying it without knowledge. I don't know. But in the mouse, we knew of the salt and pepper organization at the output level. This is known. Now I'm saying that it's also, maybe not surprising, at the input level, that the input themselves are salt and pepper, but they are salt and pepper at the level of individual neurons. This I did not know before. I didn't know anything before, but this is certainly the result. So this is the result, this mixing of inputs on the dendritic tree. So this is a beginning of understanding at least what kind of input the dendrites experience when they have a sensory input. Okay, shall we make a little bit of a break?
Yes.
Okay. So what, quarter of an hour or something like that? Let's meet at 10 past 10.
יש קשר בין היכולת ללכת לדיין?
זו שאלה מאוד טובה. קודם כל העכבר לא רואה אותו.
כן, זה
הוא חיה שלא רואה אותו. ובאמת אבל יש חיות נמוכות מבחינת זה לא בהכרח, לא בהכרח. אנחנו יודעים שבחתול יש, בעכבר יש, באדם יש. בחתול יש, בקוף, בחתול קוף גם יש, ויש גם בכל חיות של הדבר. זאת שאלה טובה, אם אני צריך כל יום בשביל לראות טוב.
איזושהי תיאוריה על למה צריך לארגן את זה?
למה אני צריך כל יום בכלל?
ארגון חזדות, כאילו היסטורית,
למה אני צריך בכלל באמת? תראי, בסוף אני צריך איזו ליגאוט של זווית. מישהו צריך לדעת שבעולם יש זווית כזאת. השאלה היא בשביל לקרוא את הזווית הזאת, אם יהיה לי קל יותר לארגן את הרשת, כך שיהיה לי סדרה של תאים קרובים, שכשהם פועלים, כולם פועלים, החיה יודעת בוודאות שזאת זווית, או שאני אעשה את המיקסינג הזה, אבל בשלב עמוק יותר, כל התאים שמגישים לזווית הזאת, קשורים חזק אחד יותר לשני, ויש מתחבא שם קולום, מתחבא שם קולום, אבל את לא רואה אותו שרק של הקולום, אבל מתחבא, יש הרבה תאים שאנשים לזה, ואנחנו מחוברים חזק, וזה מתחיל להיראות ככה, שיש שם קולום מתחבא בתוך קולום, אבל הם לא בנויים זה ליד זה, אבל הם מחוברים חזק אחד יותר ליד השני, ואלה שולחים את האוטפוט שלהם בהכרח, באיזשהו שלב שיודע שעכשיו יש בעולם את הזווית הזאת. מישהו יודע לקרוא את קבוצת התאים שהוא לא עכשיו בקולום, שהם נמצאים לזווית, בשלב הבא,
יותר קל לחבר את הדברים שקרובים אחד.
יכול להיות, אבל אפשר לפתור את זה אחרת. יכול להיות שזה צריך פתרון פחות טוב. אני לא חושב שיש בוודאות הוכחה שכשיש לך קולום אתה יותר רואה טוב את העולם. חיות יותר נמוכות, עם קולום שוות יותר טוב, בלי קולום שוות יותר טוב, עם חיות אחרות בלי קולום. זה לא ברור שהקשר הוא כזה אחד. לא, אבל יש סיבה אבולוציונית. למה התאים בחתולים לא קוראים אחד לשני. לא יודע להגיד, זה חישובי.
אי אפשר באמת לדעת, כאילו, גם יכול להיות שלחתול זה טורף, עכבר יותר, הם עושים ראייה שונה לגמרי, אז אחד מהם זכה קולום והשני אין לו.
יש איזה עיקרון מנחה.
כן, לא, היא צודקת, זו שאלה מאוד נכונה.
שאלה נכונה, היא מאוד קטנה כדי לקבל מזה איזושהי תיאוריה.
לא, כי המושג קולום שהוא מושג כזה שמתקבע קצת בגלל יובל וניוזל, שפשוט הכניס לאלקטרון וראו שלאורך כל העומק יש אותו, ברוב הממות הוא לא קיים. כן קיים בוצפים וכנראה אדם, לפי ההבחנות של האדם.
ויש דברים
בסגנון, אם תהיה לא ברזולוציה של
לא, ברור שלא, אבל בסדר שכן ראיתי רואים אזורים
שהם דומים אחד לשני, זה נכון. אז יש איזה ארגון יותר מסודר נגיד, בחיות גבוהות יותר, אבל יש גם ארגון מסודר כזה בחיות גבוהות יותר. יש עבודה מאוד יפה על תיאוריה של למה כדאי שיהיו קולום. יש עבודות פיזיקליות, אפשר בגרמניה שעכשיו פרפלט, שקודם כל משווה את כל האורגניזציה של B1 באבולוציה, בחיות שונות, אז מראה איזה פורק של קומפקטיזציה שמדגיש את היתרון של קולום.
Yesterday I met my cousin. Actually, I met two cousins. I have a boyfriend. Yeah, I mean in Tel Aviv. I have a class. So I have only one cousin that lives here, but another one lives
איך
here.
לארגן אינפורמציה?
Yeah, like subject case,
אפשר לעשות הרבה דברים טובים עם רנדום, חישובים, קצת קסקל יותר טוב עם רנדום, גם מבחינה פלסטית, שינויים. כאן נורא לעשות שינויים, כי זה כמו שאמרתי, סינפסות עבודות ולעשות שינוי מאוד מהיר ברגישות של אדם.
כן, אבל כמה יש שינוי בשלבים רוחבים?
אבל אם תחסי לחתול, שלבים מאוחרים זה נכון, אבל אם תחסי לחתול את העין שהוא בור, בשלב מאוחר יותר, לא יודע להגיד אם תמצא עצב משנה ילדים מזרחים מראה שדמעות אחרי לידה, הסלקטיביות של הקר, משתנה, גם בחידוד וגם בקטזן, כל יום אין כל העלות על פלסטים, לפחות בתור אין מולדת, בצורך לשמוע את הצעקה של הבנה. זה מקרה מאוד ספציפי, אבל כן, בסדר. או אם אתה רוצה, זה מתמחה. אם אתה נהיה ארכיטקט.
זה רמה גבוהה.
אני לא בטוח. לא בטוח. לא, אני חושב שיש הוכחות שאתה יכול לעשות חיה מבוגרת. נכון שאתה עושה לדברים דרמטיים, אתה מכניס אותה לסביבה מאוד מסוימת, מצומצמת ודורש מנה פגישות, צ'אלנג' מאוד רציניות. אז האוריינטציה של התאים כן הולך לכיוון שצריך אותו בשביל לפתור את הבעיה המסוימת. יש לי יכולת. כי אחרת אתה שואל את עצמך, למה עם כל הסינפסות שם? מה יגיד אותה נוירוביולוגים? זה אומר שהוא יכול היה להיות מישהו אחר. הוא מקשיב לכולם. הוא מקשיב לכולם ומחליט ללכת לשם. אתה. אבל די בקלות. זה לעשות פלסטים של כמה סינפסות אולי, 50. שדווקא באוריינטציה אני תחזקנה ויחזיקו לך, כאילו יש לך תשתית פלסטית מאוד נוחה. מאוד זמינה. משהו פשוט. שם אתה צריך לשמוע את כל התאים הפרסינפטיים, כולם, אם אתה רוצה להגיד מזווית אחת.
אתה לא יודע אם זה ככה או לא ככה.
לא, אני אומר, נגיד שלא, נכון. אני חושב שזה לא יהיה. אני מתפלא שאף אחד לא עשה את זה, זה לא מודע. שבחתול זה יותר קשה לפשוט פה שמיים. אבל אם זה נכון שבחתול לחתול, כי מאיפה היא מקבלת? אם היא מקבלת את הקלטים מרוב התאים הקרובים, והם דומים לסינפסים שלהם, גם הם דומים. אז אתה תבחי לחשוב, ולכן שם אתה יותר מקובל, כולם מדברים איתי בזווית אחת. להגיד לכולם לזוז לזווית אחרת זה קצת יותר קשה, מאשר לתת קבוצה שכבר קיימת. מבחינה זאת, אבל יכול היה להיות יותר פלזמי, ולא נושא יותר פלזמי. זה נשמע כאילו מנגנון טוב לפלזמי, לשינוי. מה הם עושים שם כולם? למה אני צריך להקשיב לכולם? למה אני צריך שיהיה בלגן על הדברים שלי? תשמע, אתה 45. בין פעילות. פעילות יכול לאוריינטציה. כלומר, אני קצת מקשיב ל-43 מעלות, קצת מקשיב ל-30 מעלות, כל הזמן מקבל אינפוט, בסוף מוציא אאוטפוט אחת. איזה מין חיווט זה? איזה מין חיווט? יש קצת עבודות של ישראלים יותר לשתק סינפסות עובדות, אופטוגנטי. הוא מכניס סנסור לסינפסה, ומדליק אותה ומחבר אותה, את הסינפסה, או את האקסון. את האקסון בסינפסה. שם את האקסונים אפשר להדליק או לחבר אופטוגנטי. אבל זה כל מי שמדבר איתו. אבל יש כאלה שמתחילים לשתק סינפסות ללא מדליק מאוד. העולם הזה של האופטוגנטי זה אמור.
רגע, אז לא עושה יש יותר אחורה? זה לקוח בתוך המאמר שלנו, אני מניח.
עוד פעם, סליחה? מה אמרת?
זה לקוח בתוך המאמר, אני מניח.
כן, כן, זה המאמר הזה. זה המאמר.
זה לא, לפחות לא יודע, דברים שמצאו פה, שום דבר מהם, זה לא נראה כאילו זה מחסה את כל האופציה.
לא.
רק כאילו שזה מאוד אוריינטציה חזקה למעלה עם קצת הוויות.
נכון. הוא תפס, כמו שאתה רואה, הוא תפס גם סינפסות מאוד קרובות לגופתה.
כן.
שיכול להיות שהן מכתיבות, כמו שאמרתי, שהן מכתיבות, אולי את הזווית הזאת, באוטפוט. אבל הוא מודע לזה הרבה במאמר, כי יש לו את האלקטרות בגופתה. אבל הוא לא מצליח לעשות את זה. אבל אתה רואה שיש די הרבה עם אוריינטציה בישראל, ואולי קצת זה, ואולי קצת זה, ואולי קצת זה. אז יש כאן 5 סינפסות מכיוון אחד, ואולי כמה מהכיוון הזה.
אני לא יודע מה בסוף הוא הגיע למסקנה ש...
לא, זה רק דוגמה של תא אחד.
אבל זה לא נראה כאילו כל תא מקבל באמת אוריינטציה מהכל.
זה מספיק, יש לו מספיק תאים בשביל להגיד, נכון, פר תא אני לא יודע להגיד לך.
הוא לא מחסה, זה אפילו לא נראה, זה ממש לא דגימה, כן, אם זה דוגם, זה מקבל משהו כזה, זה ממש לא נראה.
אולי אני מקבל רק 3 זוויות.
לא, אולי אני מקבל גם את כל הזוויות, אבל מ3 זוויות אני מקבל 80% מהמידע שלי.
אני לא יודע, זה בדיוק לא יודע.
בייס למשהו.
בדיוק, זה בדיוק מה שחסר לי. כי לפי זה נראה שאתה באמת דוגם קצת קבוצה קטנה של זוויות.
אם אולי את זה במאמר, זה דבר הכי...
זה הגרף הכי חשוב שלו במאמר.
אז זהו, אז אם הוא יראה דווקא את זה ולא משהו אחר, זה נראה
שזה סמפלינג.
שזה נראה שרוב הדגימות נראות ככה, או שהדגימה
רוב הדגימות נראות ככה.
כנראה שאין לו משהו שבאמת רואה את הכל.
יכול להיות.
ואז...
יכול להיות.
ואז המסקנה נשמע קצת מוזרה לנתונים.
אתה מדבר על המסקנה הזאת.
כן.
זאת, זה נראה לך שהוא דוחף את זה קיצוני, כי הוא לא יודע.
זה נראה לי, אני לא רואה בדאטה את זה.
תסתכל, קודם כל אל תשכח שהוא משתמש, אם אני לא טועה עכשיו, זה ב8 זוויות. אני עכשיו, כן, אני כן זוכר. זה הסמפל של הזוויות, כן? זה או לכיוון הזה, או לכיוון הנגדי, כלומר יש לו 8 דיירקשן. לא את כל עולם, זה לא את כל עולם הזוויות, כן? תמיד בניסוי כזה אתה חייב להגיד את זה. אז אני אומר, מתוך ה8 זוויות האלה, זאת, נגיד תא מסוים אתה רואה סינפסות בעיקר נגיד מהסגנון הזה, אבל זה מתוך הזוויות שהוא נתן. הוא לא נתן עוד זוויות לתא הזה, וראה שיש עוד זוויות אחרות שהוא נתן. אבל אתה צודק, אני מסכים איתך באופן עקרוני, אבל אני רוצה להגיד שיש מאמרים יותר מתקדמים מבחינת הטכנולוגיה, כמו למשל המאמר שלו על האודיטור, שעשו בגרמניה, ושם זה על פריקוונסיס, איך ממופים פריקוונסיס לדברית, ושם יש לו כבר 100 זוויות, ואתה רואה שזה יותר דייברס, מה שרואים. מבחינת תדרים עכשיו, נדבר על אודיטורים.
לא, אבל אודיטורי זה עולם אחר, זה הגיוני שהם יקבלו כמה דברים.
הרבה דברים.
כן.
למה שם זה יותר הגיוני? מה הגיוני באודיטורים?
כי בוויז'ואל אני כן יכול לחשוב שתא יורה לפחות אני יודע שתא יורה באיזושהי אוריינטציה מסוימת, אני לא יודע את זה על...
בתדר מסוים.
אני לא יודע אם תאים יורים בתדרים מסוימים באודיטורים.
כן. לא,
הם לא מחברים את זה, הם לא עושים איזשהו איחוד.
לא, אבל אתה יכול לעשות ריספטי פיל של תא אודיטורי, לתת כאן תדר. ואתה תקבל עקומה כזאת. תגיד שהתא הזה בעיקר רגיש ל8 קילו-הרץ, אני לא יודע מה.
אבל הוא לא... כאילו... בתדרים אני כן יכול לשחק, אבל לא רק עם התדר, אלא גם עם ה-H והזה, ויכול להיות שהתאים האלה יורים ואז מוצאים את הפורמט.
גם פה אתה יכול לשנות את העוצמה של הזווית, את העוצמה של הקונטרסט. גם פה יש לך עוד פרמטרים, כן? אני יכול לתת את האוריינטיישן הזה בקונטרסט אחר או במהירות אחרת. יש המון פרמטרים ויזואליים. כאן הוא עושה תמיד במהירות של, אני לא זוכר כמה שנייה, הוא מזיז הוא לא עושה עוד, כי יכול להיות שהתאים האלה, אתה צודק, יכול להיות שאם הייתי משנה את מהירות התנועה של הזווית, או את הגריידינג עצמו, את המרווח, הספיישל גריידינג, הייתי מקבל התנהגויות אחרות. זה מאוד פרלימינרי, אבל כאן הוא לוקח קונטרסט מקסימלי במהירות קבועה, אני חושב שזה 1 ל2 שניות או משהו כזה, עם כזאת צפיפות של גריידינג, וזה מה שהוא מתאר. למרות זה מאוד לימיטד. זה כמו במערכת השנייה.
אגב, זה איזשהו בעיה... כאילו זה איזשהו, משהו כאילו יש משהו מתוך הניסוי שמכריח אותו לבחור רק סינפסות קרובות לאסור?
כן, יותר קל לו מבחינה אופטית, כנראה לאתר אותם. אני לא עד כדי כך מזהה את ה... תראה, הוא מזהה את גוף התא עם אלקטרוד, לא מכניס לשם אלקטרוד.
כלומר, אם הוא היה יכול, הוא היה בכל הדברים שמקים.
ברור. הוא לוקח אלקטרוד, ועכשיו הוא מתחיל לחפש, הוא מסמן את גוף התא. שם יש לו אלקטרוד.
הדברים הצהובים האלו זה דברים שהוא מנחש?
לא, זה דברים באומתים שם. הוא יודע שהם שם, אבל הם לא נמצאים בפוקל פליין הזה. בפוקל פליין הזה זה נמצא בפוקוס, זה נמצא בפוקוס, וזה נמצא בפוקוס. אבל הוא יודע שהם ישנם, כי בפוקל פליין אחר, אז הוא חוסק את הדברית הזה, כן? הוא מחפש ליד הסומה, הוא נמצא שם עם אלקטרודה, כנראה שמשום מה, אני לא יודע בדיוק למה, הוא סורק עם התופוטון בסביבות של הסומה, אבל אין שום סיבה כמובן לא ללכת להפך, רוב הסינפסות נמצאות רחוק, אני לא יודע עד הסומה. אבל הוא הלך לזה. אבל כמו שאמרתי, זה היה הראשון בסדרה של מאמרים.
הוא יכול גם לבדוק מה אינהיביטורי ומה אקטיביטורי, או
לא.
שלא דרך לדעת את זה?
זאת אחת המכשלות המאוד גדולות פה, כי הסיגנל שלו זה קלציום. מה שהוא רושם בתרופות במייקרוסקופ זה קלציום. הוא מקווה שזה ברמה של בנקודה פוסט-סינפסית, ממש מתחת לסינפסיה, או מגבול לסינפסיה. אבל בשביל לקבל קלציום זה תוך דפולריזציה. לפתוח, לעלות, דפולריזציה. הסינפסיה האינהיביטורית לא מכניסה קלציום. אז היא פועלת בטוח, אנחנו לא רואים אותה. בטכנולוגיה הזאת אתה לא יכול לראות סינפסות אינהיביטוריות. פשוט לא יכול לראות. אתה כן יכול לראות סינפסות אינהיביטוריות אם אתה מתחיל לעשות איזו היפופולריזציה מאוד חזקה או משהו מהסוג הזה, אבל אתה צריך שהסינפסיה עצמה תעשה מספיק לוקלית, דפולריזציה בשביל לפתוח תעלות קלציום. אבל אני מבין שלא עושה את זה. אז אתה תקוע.
אנחנו לא יודעים אבל שגם הפיד, כאילו, נכון יש שנותנים לצום לזה שפגע כאילו זרם פידבק חזרה, שהוא גם פתח תעלות קלציום.
אתה מדבר על הספייק החוזר.
כן.
בדיוק.
זה לא ממש מוסיף.
כאן הוא רוצה, במאמר הזה הוא טוען שאין לו, זה היה לא רק מוסיף, זה היה עושה מסקינג של כל הדברים האלה. הספייק החוזר, שהוא קיים, הוא מכניס כזאת כמות של קלציום, שהוא שורף את כל הקלציום הלוקלי מבחינת כמות. אבל כאן אין לו, הוא דואג גם שלא יהיה לו, בין השאר בזה שעושה היפרפולריזציה. הוא עושה היפרפולריזציה לגוף התא. ודואג שלא יהיה ספייק בתנאים מסוימים.
אז איך הוא מודד את זה?
לא, זה בדיוק הדרך שלו לחלק. אתה אומר, אני לא רוצה שהתא שלי יירה לרגע. לרגע, לא כל הזמן.
אתה אמרת שהוא בודק מה...
לא, אני אומר לרגע, לרגע אני רוצה שהוא יירה. עכשיו אני רוצה רק לראות את הפלוס. אז אני עושה היפרפולריזציה לתא הפרוסינפי ודואג שלא יצא בו ספייק. לרגע. ואני רק מאפיין עכשיו את הקלטים. הם ממשיכים לפעול. אני לא נוגע בהם, אני רק מקלקל את הפלט שלי.
ואז אתה מודד פלט?
מה?
ואז אתה מודד פלט?
ואז, אחרי שאפיינתי את הסינפסות, כמו שאמרתי, זה שדאגתי שלא יפריעו לסיגנלים אחרים, אני מבודד את הדבר הנקודתי, את האינפוט, עכשיו אני נותן מתי לפעול, גם הוא, אני משחרר אותו מהיפרפולריזציה, ואני מאפיין את הפלט שלי. עכשיו, לא קלקלתי את הפלט, כי אני כבר לא רושם עכשיו קלציום, אני מפריד.
אתה רושם את זה בשני מצבים שונים, אתה לא יודע, בהינתן הקלט הזה, אתה לא יודע מה הפלט שיצא לו, אבל אתה יודע מה הפלט האופייני.
אני קודם כל מאפיין את הקלט על ידי זה שאני מבודד את הפלט, ואחר כך אני נותן לפלט הרגיל, אני אומר, אתה זה 45 מעלות, נתתי לו לפעול רגיל, ואני גם יודע משהו על הקלטים, כי הנה קודם רשמתי אותם כשלא היה פלט, הם נקיים נקרא לזה לחצים. זה הטריק, זה החצים שלי.
כאילו, שני דברים רק אני אשאל אותך זה עוד רגע. אתה אמרת שמטרה שלכם נניח לחשב את הפונקציית פעולה של
קלט פלט של התדריך.
למה, כאילו, ושחקן אינפורמטיק לא מודדים בלי אבל למה...
זה לא תמיד, אני באמצע שיעור, אני יכול לעמוד שעה.
למה מחקרים שיש לך מיליון על בסיס נניח ואתה פשוט יכול לשלוט בכל אחד מהאינפוטים וזה, אתה לא יכול בקלות לקבל בכל הדאטה שלך?
זה בדיוק דברים שעכשיו עושה למשל ירום, מישהו איטלי, פיזיקאי, שהוא זה לא קל להפעיל סינפסות אינוויטרו על הצ'יפ, זה לא קל להפעיל את הסינפסות עצמן, אחת. מה שאני אומר,
למה ננו אלקטרוניקס כאילו מגיעים, אפשר לעשות שיפים ברזולוציה?
לא, אבל איך אתה תפעיל, נגיד שיש לך תא על הצ'יפ, תא גדל לך על הצ'יפ, פלייס שנמצא על הצ'יפ, אתה יכול נגיד לרשום את הפעילות מהצ'יפ, אתה יכול לרשום את הספייקים של התא הזה. אתה לא יכול לרשום את האינפוט הסינפסות היום, אין לנו רזולוציה של לרשום סינפסות בודדות. בצ'יפ, רק האופציה הזאת מאפשרת לנו לראות פעילות של סינפסה בודדת היום.
אבל כאילו לא צריך לראות, אתה יכול במחשב לשלוט ישר במה שלא יש לך איזה אלקטרודה שם קטנה ותגלה אותה חשמלית או משהו.
לא, אז כן, אז מה שעושים היום, מה שצריכים לעשות זה באופטוגנטיקה לעשות לגרות, בוא נקרא לזה להטיל תמונה לגרות על ידי תמונה של אינפוטים, נגיד את המחלום של פה ושם יהיה לך אתה מדליק את התאים האלה אופטוגנטית, אתה מפעיל אותם אופטוגנטית ונשאר מהקפסות. אתה לא יודע איפה משודרים עליהם. אתה לא יודע בדיוק שאתה עושה טק על הרשת, ואתה רואה כזה, אתה לא יודע איפה סינפס.
אבל זה אפילו שיש לך רשת, אם יש תא בודד,
אם הוא בודד זה לא סינפס. אבל אם הוא בודד זה דרך אחרת לעשות את זה להפעיל לוקלי, איכשהו חשמלית לגרות פה על הדמרית. מתחילים לעשות את זה, ואז להביא את יחסי הקלט פה.
כאילו אותי זה אופן כלי נורא שקל של כמעט לא שומע על מאמרים של מגדל רשתות אינוויטרו.
יש עבודה בין שמעון ארון בטכניון, שמעון מרום עושה את זה בטכניון, ויש בחור בשם אלישע מוזס, שעושה את זה בוויצמן. אלה שני הישראלים שמגדלים על צ'יפ. רשת, לא תאים בודדים. אני חייב להמשיך. Okay, let's continue. Okay, I just want to complete this part by showing you so the technology that enables to record from dendrites the input itself is also being applied by another group from Germany, Axel Borst, and a postdoc in my lab working with him and with me, Siwei Wang from China. She's trying to understand this, in this case it's a visual system of the fly. A particular region, there are these beautiful cells that compute, you know, the fly is extremely good in maneuvering in the world, using very fast visual input, and they can do this amazing thing that the flies can do, you know, without going into the wall and so forth, following another fly, chasing another fly. And these are important cells, very few, only 10, that are responsible for the vertical orientation of the visual orientation of the fly. So we are trying to understand, in this case we have the location, also calcium imaging, that's the technology today. Not two photon in this case, but calcium imaging from this region, and you can see how the visual world is mapped also on this particular cell type. So, you know, region number 1 in the dendrite is very sensitive to this orientation. In this field, and point number 7 is sensitive to this orientation in the visual field. So its location and orientation are mapped into the dendrite, and eventually there is an output on the axon. And so here we have a very good chance, and we are approaching to at least understand this cell in terms of its input-output. But luckily it's a relatively easy cell to record from, also from the dendrite. It's thick, and you can record with an electrode here and visually there, and we can really characterize. I mean, when I say we, I mean this is a group of wonderful group of Axel Borst from Germany. So we are trying at least to understand the input-output properties of these VS cells, the vertical system cells in the fly visual system. But I don't want to go into this particular work. As I said, I have a very limited time, and I could all the talk could go on only to this paper that is about to be published. So I don't want to go there. I just want to mention what we are capable of doing today in terms of input-output recordings in vivo during some sensory processing from dendrites and axon. That's what I wanted to give you. Yes, what happens today. Okay, so I want now to go to cable theory. To what we do understand from the cable theory about dendrites, at least on the passive regime, on the linear regime. And we'll talk about nonlinear regime. So you have all my lecture in the Moodle, so you can look at it, and I don't want you, of course, to remember everything at all. I just want to give you some key numbers for you to remember, some key parameters for you to remember. So if I look at the morphology of a cell, of course it's very diverse. I cannot say that this is the dimension of a neuron, but I can say something on average. So let's look at cortex. It's nice to talk about cortex, but we talk about the cerebellum. Let's talk about cortex. If Hagai Bergman was here, he said, why do we talk about cortex all the time? Most of the cells are in the cerebellum. He's right. 80% of the cells in the brain is the cerebellum. But we like to talk about cortex because we feel that we are cortex in many ways. Of course, we need a cerebellum. So I'm talking about the cortex, just as an example. Okay, so just to give you a ballpark, it's not hard numbers, it's average numbers. So let's say, what is the diameter of a dendrite? Average diameter of a dendrite. It's 1 micron. It could be less, but it's around 1 micron. Okay, that's the diameter of a dendrite. The axon is thinner. It's harder to record from axon electrically because the axon is thinner. It could be 0.1 micron. So dendrites are rather fat relatively, although it's very hard to record from them, and they're also short. They can extend, let's say, to 1 millimeter. So an axon could be 1 micron in diameter, 1 millimeter in length. Not total length, but the merchak aviri, from the soma to the tip on average is, let's say, 1 micron, 1 millimeter. Okay, so these are good numbers to remember. Could be sometimes more, if I open all the dendrites, if I just make all the dendrites and compute the total length, it could be several millimeters, 10 millimeters, 20 millimeters. It has a lot of area. There are small cells with only 2,000 square microns, but there are cells which are very big, very expensive, like Purkinje cells. This is not cerebellum. This is in the cerebellum. Could be very, very big. So actually, if you ask me in terms of area, who has the most area in your brain in terms of membrane area? So it's the dendrites in the cortex is about 60% of the area. And membrane area is in the dendrite. And although you have many, many, many wires that are called axons, because they are so thin, totally they may be 40%. So let's say 60-40. 60% of the area is in the dendrite, 40% in the axon, and 1% or less in the soma. So the soma is really, really lucky for us. You can record with an electrode, but in terms of area, very, very small. Most of the area is in these branches. So we are a branching system, not soma. Of course, there are several dendrites emerging from the soma. In the Purkinje cell, there is only one dendrite. Purkinje cell is a very unique cell. From the soma, there is only one dendrite, and then branch. But in the cortex, all the cells have more than one dendrite coming out, and each one forms its own dendritic tree. There is a basal dendrite with 7 dendrites or so coming out. There is an apical dendrite with 1 coming out and then branches. But totally you have many, many stem dendrites coming from the soma. So this is morphology. Of course, we will speak about spine, and we should speak about spine. I will show you later that Ramon Cajal, just before he died, really just before he died, as he was, as I said, this graphical maniac, he wrote, and I'll show you later, he just wrote, look at the spines. These are not artifacts, because people thought that these little things that are coming from the dendrite are artifacts. They are very, very important. And he died. And we are still studying what is this spines for, but we'll talk about spines later. So I can speak about morphology, and that's what Ramon Cajal tried to do, but today we can speak about physiology. And in the physiology, when I speak about physiology, I mean electrical properties of the dendrites. So we can speak about passive properties of the dendrites, which I hope you know. You got some initial course about passive, isn't it? So I don't want to go fast, but you have this membrane resistivity, Rm, and this is the units. It's in ohms centimeters square. You have the axial resistivity of the cable, of the dendrite, or the axon. This is Ri, axial resistivity, or R internal. This is in this unit. And of course, you have the membrane capacitance per unit area, Cm, which is typically 1 microfarad per centimeter square. So it's a universal number. Until we recently found that this is not universal in human. But we should talk about human later, maybe, if I get to it. So anyway, these are the 3 parameters you need to characterize the cable, the electrical properties of passive cables. All these parameters are passive, so they are static. They are not voltage dependent. They are static, so these are the numbers, and these are the range of numbers. For example, just to make it clear, let's say that I'm asking what is the time constant of the membrane, which is an electrical parameter of the membrane of neurons. It's Rm multiplied by Cm, and if you multiply these numbers with these numbers, you get the time constant, which is an essential, absolutely important key parameter to understand neurons. The time constant is on the order of tenths of millisecond, 10 milliseconds, 20 milliseconds, 30 milliseconds, something like that. Okay, so this integration time constant of the membrane is on the order of tenths of milliseconds. It's a very slow clock in terms of the time constant inherent to dendrites, to neurons. I can speak about how long are dendrites electrically. So I don't know, you remember the term lambda? The space constant of a neuron? Okay, so I can ask in terms of units of lambda, not in terms of anatomical units, because I said anatomically it's 1 millimeter. But I can ask how long electrically, in units of lambda, how long is the dendrite in units of lambda. I can say that it depends, of course, on how long the dendrite is, but let's say the apical dendrite in the cortex of pyramidal cell. The apical dendrites could be 1 lambda long, 1 space constant long, and the basal dendrites, which are shorter morphologically, also shorter electrically, it's let's say 0.3 lambda. So that's the range of the electrical distance. We should talk about this in a second. Why is it so important? The range of electrical distance of neurons is lambda, on the range of lambda. And it has, of course, implications for attenuation of current, voltage, charge, and so on. So you can look about these passive properties, and you can, so attenuation and so forth, we'll talk a little bit, and then you can, of course, add on top of it, which we know more and more today, actually, that the dendrites are absolutely rich with a soup of ion channels, non-linear ion channels, voltage-dependent ion channels, rich. Actually, the richest place in the brain in terms of usage of ion channels, different ion channels, is the dendrite. So it's not passive, it's not fully active, really. We'll talk about activity, non-linear activity in dendrites in the next, maybe, I hope, in the next, maybe even today. So we have to also characterize, of course, what are the type of ion channels in the dendrites. I want to mention, to remind you something that you know. There are only 4 types of ion channels in general, basically. Calcium ion channels, sodium ion channels, potassium ion channels, chloride ion channels. Basically, there are only 4 ions that flow through the membrane. So when I say I have many ion channels, I mean all these many ion channels are transmitting current of these 4 types of ions. But I can say that we know today of 50 potassium channels. 50 different types of potassium channels. All of them transfer potassium from inside to outside when they are open. So it's only potassium. But when I say 50, I mean that they have different time course. So the time course of the channel is different. Some of them are very brief, some of them are very slow. So there is a time course and there is the voltage dependency. Some channels open in hypopolarization, some channels are open in depolarization. So there is this different sensitivity to time and voltage. And sometimes also to another ion. So we know, for example, there are potassium channels that are sensitive to calcium. So when they see high concentration of calcium inside, they open. Okay, but it's only these four ions that play between themselves, and the differences are in time and in voltage. And there is a big effort today, especially with Henry Markram, we should talk about him on the Blue Brain project, but he has this whole project, it's called Channelpedia, the encyclopedia of channels, meaning that I want to find out which part of the genome codes for this type of channel. It's key, you understand that the activity of neurons absolutely completely depends on these channels, also of the heart. So if I have a heart problem in terms of beating or pacemaking, it's some channel that doesn't work, potassium channel. So these channels are key for understanding electrical activity in the brain, of course, and diseases. So we need to know what are these channels and what are they blocked with and what are they open and what is this mix. Why do we have all these channels that just make life difficult, really difficult? Because all these channels can change the relative the densities and they have different activity because of that. Sometimes you have more potassium and it blocks the cell, and some less potassium and the cell becomes epileptic. So these channels are problematic. There are so many of them. Why do we need so many genes to code all these channels, and most of them are in the dendrites? It's not so clear, but it's a fact. Yeah. The physicists will tell you it's just noise.
Yeah,
it's a biological noise.
Yeah, I'm going to give like...
It's a biological noise. We don't care.
First you said it's like you need to understand the neurons, to understand the functions, the higher level functions. Okay, I can accept that. But then, now you say that you have to differentiate 50 types of potassium channels, each from another, to understand how the cell functions, so you can understand a higher level pharmaceutical. I mean, next you can say that you need to know every single protein in the and atom in the...
Biologists will tell you exactly that.
But this, like, everyone will agree that it's like impossible to do now and will be impossible to do in like forever. Like you can prove it computationally that it's impossible. So, question is,
do you have a sort of a proof or a reason to believe that the 50 types of channels really do play a major role in like higher level functions? Like if you knock out one specific type of channel, the organism won't will behave differently? Or?
Okay, that's a fantastic question, and that's the whole issue about what do you need to understand in order to understand the nervous system at a certain level. Of course, if you speak about diseases, if you are interested in diseases, absolutely you need to understand these channels, because you know, as I said before, that some of these channels that we know that have some genetic issue with this channel, you have a severe disease. One channel. So for this part, of course, physician will say, of course we need to know the channel and also physiology. Some people say, okay, this low resolution, detailed resolution. On average, we don't care about 50. Let's say there are 2 big ion channels for potassium. It's enough to understand basically all the activity. And I will agree with that. But this is the question of the high-level question of when I talk now, when I do some computation. But this is an issue. I'm just saying that people are, because we are interested in the characters and we want to understand everything, and you have tools, and you want to understand all these channels. Then you want to block this one and this one and this one, and you want to see how it shapes the output. I'm just saying that this is the direction. But this is a very good point. How low should I go in order to understand high-level computation? That's why in this center we have physicists that do not like first the connectomics, but we don't want all this. We need a little bit statistics and something like that. The biologist said, okay, you didn't show us that you understood anything until today. Let's tell me, I will do what I can do, and then show me that it's important or not important, but at least you know what exists. Something like that. This is the type of argument, but it's not as deep. It's not very deep what I just said. I just said that we don't know what is important, so we explore what we can explore. I'm just telling you what we know. My role is now to tell you what we know. Then we can discuss later if it's important or not. Of course, we have to know also the synaptology. So we have to do morphology and physiology, something at some level, and we need to know the synaptology. Where are the synapses? Where are they coming from? Are they inhibitory? By the way, I was asked a very good question before in the intermission, which I should have said. In the paper that I showed you before by Connors, about this visual input, and the synapses, these were only excitatory synapses. This is important. So I don't, with this method of optical imaging, calcium imaging, I cannot see inhibitory synapses because they don't generate depolarization, and so they don't activate calcium channels, or NMDA channels in this case. So I don't see the inhibition. And of course inhibition plays a key role in shaping the output. But this technology does not allow me to see the activity of inhibitory synapses. So I cannot say anything about that. I only know the numbers. I know that the pyramidal cell gets 90% excitatory synapses and 10% inhibitory synapses in numbers. But I don't know exactly when they are active and so on with this technology. So I need to know synaptology, and I need to know, to tell you, of course, that there are these 2 types of, the key types, the important types of excitatory synapses, those that carry NMDA, that have the NMDA receptors, or AMPA receptors, non-NMDA or AMPA. They sometimes are called non-NMDA or AMPA receptors and the NMDA receptor. And both receptors are sensitive to glutamate in the cortex, so the transmitter is glutamate for both receptors. The difference in these receptors, of course, is this receptor is a very interesting receptor because it depends on voltage. Unlike the classical receptor that only depends on the transmitter itself, the presence of the transmitter, NMDA depends both on the presence of glutamate and on postsynaptic depolarization. Only then the receptor is open. And this is the basis for plasticity, coincidence detection, and this is a very important receptor that was found some 25 or 30 years ago in the nervous system, in the cortex, in the hippocampus as well, not in the cerebellum, for example. And there is the inhibitory synapses, which also have basically 2 types of receptors, the GABA A and the GABA B, so both are sensitive to the GABA transmitter, but they have 2 types of receptors for the same transmitter. This is opening chloride ion channels, and this is opening potassium ion channels. This means that this GABA A do not check, because chloride is on the reversal potential. The battery of chloride is near the resting potential. This synapse on its own do not change voltage. It's called the silent synapse. Only when it's on the resting potential. When you shift from the resting potential, when this synapse is active, you have the hyperpolarization if you depolarize the cell. But this is one receptor which carries chloride ions, and this is the other receptor which hyperpolarizes the cell because it opens potassium channels. And potassium reversal potential is negative relative to rest. Okay? So that's a quick summary of synapses. So let's speak a little bit about cable theory, okay? So here is another controversy. So this is John Eccles, Nobel laureate. Dendrites are not important. This is Wilfried Roll, who was his student. He's the father of dendrites in terms of modeling. And also my postdoc supervisor, a very special person really, a theoretician, physicist. They really fought a lot. And this guy, you can see how mean scientists could be, selfish, mean, and idiotic, and think that they know everything a priori and so on. So he was his supervisor, but he made sure that the paper, the most important paper on cable theory, the most important paper, a big paper that he wrote for many years, mathematical paper on cable theory, he didn't want this paper to be published, and he was Nobel Prize, so he told the editors, this is nonsense, dendrites are not important, it's a waste of time. But luckily for Wilfried Roll, he found one paper that was just open, experimental neurology. Nobody heard about this paper. Even today nobody knows there is a paper, a journal called Experimental Neurology. He found this editor, and this paper was published there. The most important paper in cable theory was published on a journal, sneaked into the journal because the Nobel laureate thought that dendrites are not important. Not only that, but because cable theory showed that he was wrong in many of his interpretations. So now we are going to why models are important. Okay? But you can see that there is behind, of course, your human, complicated characters, complicated brains in many ways, also politically. We can talk about this later. We are doing bad things to ourselves here in Israel, and not only here. We vote for Trump, for example. So what is cable theory? So as I said, this was the concept, even until today, that neurons can be well represented as points. A point neuron, one compartment neuron. It's a good representation. Why do we need all this complex tree? It's not important. Actually, what Eccles said is the following. He said, I know that there is this anatomy. Here is the beautiful tree that you saw, the most well-known tree that you saw by Ramon Cajal. So Eccles, of course, knew about this anatomy. But he said, okay, the distal synapses that are here, are essentially not effective. So we ignore them. Nearby synapses are effective, so we put them on the cell body. So as a first approximation, we ignore distal synapses because they are distal, and we take proximal synapses and we put them on this model. So this is my model. This was the argument. Why to neglect dendrites? Why dendrites are not important? This was his kind of intellectual argument. But of course, Roll and everybody else knew that this is the biology of neurons. So what does it mean? Is it functionally meaningful, or maybe it has another reasons to have such a complicated tree, but functionally you can really replicate most of the interesting activity of a nervous system, of a network, by a point neuron. Sometimes it's called McCulloch and Pitts neuron, sometimes it's called a point neuron, but I mean to shrink the whole dendritic tree into a functional point of summation. That's what Eccles did. But Roll showed in this paper from '59 some of the things that he showed I want to highlight now, because this has changed completely the way we thought about neurons, at least biologists or computational biologists that cares about dendrites like myself, and this was his reasoning, the Roll, also mathematically. First of all, let's say that you're in a cell body, here is your electrode, and you record from the cell body, which is what we knew to do until recently. So you record from the cell body, and Roll was just saying something extremely simple that you can, of course, prove mathematically. But you know it now, because the cell body membrane area is so small relative to the membrane area of the rest of the dendrite, is that when current is injected to the cell body, or when current arrives from the dendrite, from the synapse, when there is charge coming here, most of the current that is injected to the cell body either by an electrode or by a synapse that is coming from distally, most of this current escapes elsewhere. It doesn't go through the membrane of the soma itself, but it goes elsewhere. Because most of the resistance, I should say most of the conductance is in the dendrites, not in the soma, just because of the membrane area. Okay, so if I inject the current here, very little of this current will go through this membrane, very little of this current. Most of this current will escape, will be synced to the dendrites. So this is one intuition I wanted to give you. I'll show you mathematics a little bit later, but this is enough to say that because the soma conductance is relatively small compared to the conductance of the rest of the membrane, so most of the current, doesn't matter where it comes from, will not charge your membrane of the soma, it will go elsewhere. It will escape elsewhere. So that's one intuition, very basic intuition. Okay? So the question, of course, because of this intuition, we'll talk a little bit about that, how good is my approximation when I take a real dendritic pyramidal cell, in this case with apical tree and with basal tree, and I shrink it to a point neuron? What do I lose, if at all? So because Eccles thought that this is a good representation of this cell, his interpretation of the signals that he recorded from the cell body, so he recorded from the cell body of this cell, but he interpreted his model in his brain was it for an isopotential point neuron. He made huge mistakes in the interpretation of the data. The data they interpreted, the experimental data, was misinterpreted because of the model that he has in his head. And that's what I want to show you in a second. What was the problem? So, of course, the question is, and that's an ongoing question, including in my lab, do you want all the details to understand high-level function? Can you reduce it to this level, or maybe there is some intermediate stage of reduction, theoretical reduction, of this complexity maybe to this complexity, that you can capture most of the important computational things that the cell is doing, maybe not with this compression or simplification, but rather maybe with this simplification. Is there a simplification that is good enough to capture the complexity here? That's a question. Okay, so let me show. So for me, it's doing the following. Yes. So this is the reality. It's still a model, of course. You have a model of a tree. It's never with all the details that you mentioned just before. It's not. All the fine details are not there anyway, so it's a model. But this is the model of a kiss. But it's a detailed model. It's a detailed model because you have a detailed kiss, and you have a body, and you have legs, and all these hands, and so forth. And there is another model of a kiss. This is the model of the kiss here by Brancusi. Both of them are in Paris, of course. So you have, this is the city of kisses. So you have to go to Paris to see Rodin Museum or Brancusi Atelier near the Pompidou Center. So these two kisses are in Paris, but this is the high-level model of a kiss, of course. It's a reduction of a kiss to the minimal, yes? Seriously, I am serious now. This is the reduction of a kiss to the minimal, because if you reduce more, you lose the kissiness. There is no kiss, yes? So if you reduce more, maybe this is a reduction that you lose the cell. You lose the properties, essential properties like a kiss, yes? So, but this is still a kiss, a wonderful kiss. Brancusi is absolutely genius in his, so he was a theoretician. He was a physicist. He liked to reduce things and capture the essence. He used to speak. He said, I want to capture the essence of the kiss. You know that he has a wonderful head, a minimal head. He has an amazing bird. That is a minimal bird. So he was a minimalist. He was a reductionist. But this is, of course, a wonderful model also. Yes, a matter of your taste, but also to explain the kiss. Is that a good explanation or explanation? What is an explanation? So this is my little metaphor because I like this. I like art and I like these kisses and so on. Okay, so so that's one aspect about reduction, and of course there is the issue of why do I need a mathematical model in biology, and this is a good start, because Lord Kelvin was a physicist. William Thomson was called Lord Kelvin, who was a physicist, and he said the following: I'm never content until I have constructed a mathematical model of what I'm studying. If I succeed in making one, I understand. Notion of understanding. Biologists will not like this, because of course they feel, and they're right, they understand something by reconstructing, by talking about this, by making a graph. It's a certain understanding. So the notion of understanding is a very complex. The notion of understanding is something very difficult to agree on. There are many levels of understanding, but at least he, as a physicist mathematician, he said, I understand if I make a mathematical model. Otherwise, I do not. I do not understand without mathematical model. So I want to give you some examples of understanding and why do we need a model. So very fast, I think, and I'll talk a little bit about that, because if you don't have the correct model, you may misinterpret your data. You see some data, you want to understand this data, where it comes from, you have a model. If the model doesn't fit reality, you just misinterpreted the data. So the model, it could be very useful if it's a good model for data interpretation. That's what I want to show you in a second about the misinterpretation of Eccles of his own data. This is why I didn't want the publication of Roll, because the Roll model showed that he misinterpreted the data. Of course, a good model, and that's what we seek for, is to capture the essence, the essential parameters that dictate this particular behavior. You don't want all these 50 channels maybe, or all these, I don't know, 100 parameters to capture a phenomena. You want to reduce it. But what are the key parameters? You don't know in advance what are the key parameters. You want a model that captures the essence with several key parameters, like in the case of Hodgkin-Huxley model. The key parameters is these 4 equations that have certain number of parameters, and this explains very well the model, the spike. Without Hodgkin-Huxley, we did not understand the spike. We saw the spike. We measured the spike. We knew that it depends on potassium and sodium. We knew many things, but we didn't have a compact model with only few key parameters. So that's another important thing about a model. Not only interpretation of data, but also highlighting the key parameters that explain the phenomena. Only, hopefully, few, not too many. And then that's the big jump. The big jump is to use a model like this to a higher level. It's not only describing a spike, but describing the computational function of spike. If a model is good, it can do the 3. It's very hard to do the 3 at the same model. From the interpretation of experiments to getting key insights to the level of computation, which is a highly different type of language. It's not a biophysical language anymore, it's computational language. But let me only tell you something about this Ramon y Cajal, and then we should stop here. What he said about us, theoreticians. There is a very nice book by Ramon y Cajal. As I said, he was very, very special person, very opinionated, like many scientists to stand and say, I know everything and so on, but he knew a lot. He said, theorists are highly cultivated, wonderful, endowed minds whose will suffer from a particular form of lethargy. We are lazy, these theoreticians. They think all the time. They don't do what should be done, study the system. So he said many interesting things. Not only that, I just picked from his book bad things that he said about us. It's amazing, by the way, that he said these terrible things about theoreticians, while at the same time, Einstein developed in 1905 these amazing theories about the photoelectric and other properties of universe, and he was saying this. Basically, that the theoretist is a lazy person. And he knows to write. Yes? He unconsciously obeys the law of minimal effort. It's wonderful to say. Because it is easier to fashion a theory than to discover a phenomenon. It's very nice. So I like Ramon y Cajal. He was completely wrong. But I cannot avoid it. That's what he said. Yes, he said also many things that you should read about how to become a scientist. Avoiding this part, but you know, he has this very wonderful idea. So just to tell you that some of these great men are saying nonsense, because we know that we understood the universe from theory. I'm not saying about neuroscience, I'm saying about physics in general. And he was, okay, and also I should also summarize that we absolutely should be lazy. It's good to be lazy. To, you know, not to do things all the time, to go, I don't know, in the beach, to be lazy, because that's where you can maybe think and maybe have some creative ideas and so forth. So I don't agree with him on a lazy as if lazy is something bad and all that, but at least I should tell you that I don't agree with everything that Ramon Cajal did, but I still adore this capability to reconstruct the nervous system from a very low level microscope. So let's meet. I don't remember, when do we meet next time? Can you remind me?
No,
Oh, that's Purim, that's the problem. So, Yom Hamishi. Next Thursday, 9 o'clock.